Good morning. Myself Dr. Ashok Siroya, Associate Professor, Department of Zoology, SPC, Government College, Ajmer. Today we will discuss about disaccharides and polysaccharides in the topic structure of carbohydrates. Disaccharides form when two monosaccharides join together via a dehydration reaction, also known as condensation reaction or dehydration reaction or synthesis. In this process, the hydroxyl group of one monosaccharide combines with the hydrogen of another, releasing a molecule of water and forming a covalent bond known as glycosidic linkage. For instance, the diagram below shows glucose and fructose monomers combining via a dehydration reaction to form sucrose, a disaccharide we know as table sugar. The reaction also releases a water molecule not pictured. So, the glucose and fructose combine with a glycosidic bond and form another sucrose molecule. This is disaccharide. So, we can say the two monosaccharide molecules combined by the bonding known as glycosidic bonding, or we can say 1,2 glycosidic linkase. These are the examples of disaccharides. This is maltose, lactose, sucrose. These are common disaccharides. In some cases, it is important to know which carbons on the two sugar rings are connected by a glycosidic bond, and each carbon atom in a monosaccharide is given a number starting with a terminal carbon closest to the carbonyl group. When the sugar is in its linear form, this numbering is shown for glucose and fructose. Above, in a sucrose molecule, the carbon number 1 of glucose is connected to the carbon number 2 of fructose. So, this bond is called as 1,2 glycosidic linkage. Commonly, disaccharide includes lactose, maltose, sucrose. Lactose is a disaccharide consisting of glucose and galactose and is found naturally in milk. Many people can't digest lactose as adult, resulting in lactose intolerance, which you or your friends may be all too familiar with. Maltose or malt sugar is a disaccharide made up of two glucose molecules. The most common disaccharide is sucrose, which is also known as table sugar, which is made up of glucose and fructose. When a long chain of a monosaccharide linked by a glycosidic bond, the structure is known as polysaccharide. The chain may be branched or unbranched and may contain different types of monosaccharides. The molecular weight of the polysaccharide can be quite high, reaching 1 lakh Dalton or more if enough monomers are joined. Starch, glycogen, cellulose, and chitins are some major examples of polysaccharides important in living organisms. Polysaccharides are stored in our body as well as other structural polysaccharides is also found in our nature. The storage polysaccharides are stored as a reserve food material in the animal or in a plant. Starch is the stored form of sugars in plant and is made up of a mixture of two polysaccharides, amylose and amylopectin, both polymers of glucose. Plants are able to synthesize glucose using light energy gathered in photosynthesis, and the excess glucose beyond the plant's immediate energy need is stored as starch in different plant parts, including roots and seeds. The starch in the seeds provides food for the embryo as it germinates and can also serve as a food source for human and animals, which will break it down into glucose monomer using digestive enzymes. In starch, the glucose monomer are in the alpha form with the hydroxyl group of carbon 1 sticking down below the ring, and they are connected primarily by 1,4-glycosidic linkage in which carbon atom number 1 and 4 of the two monomer form a glycosidic bond. The two forms are amylose and amylopectin. Amylose consists entirely of unbranched chain of glucose monomer connected by 1,4 linkage, and the amylopectin is a branched polysaccharide, although most of its monomers are connected by 1,4 linkage. Additionally, 1,6 linkage occurs periodically and result in branch points. Because of the way the subunits are joined, the glucose chain in amylose and amylopectin typically have a helical structure as shown in the diagram below. This is amylose structure, and the 1 and 4th carbon number atom of all adjoining monomers are linked with a glycosidic bond, and this is amylopectin. This is also joined with 1,4 linkage, but somewhere this is 1,6 linkage also present in this structure. Then we can say this is amylose structure and this is amylopectin structure. The glycogen is the storage form of glucose in humans and other vertebrates. Like starch, glycogen is a polymer of glucose monomer, and it is even more highly branched than amylopectin. Glycogen is usually stored in liver and muscle cells whenever blood glucose level decreases. Glycogen is broken down via hydrolysis to release glucose monomers that cells can absorb and use. We can see in this figure the amylose has a linear structure and it's made up of glucose monomer connected by 1,4 glycosidic linkage. This is a linear structure. Bottom, the amylopectin has a branched structure. It is mostly made up of glucose molecule connected by 1,4 linkage. This is 1,4 glycosidic linkage but has glucose molecule connected by 1,6 linkage at the branch point. This is a branch point. We can see here, this is the branch point, this is the branch point. The branch point is linked by this chain by 1,4 1,6 linkage at a branch point. The structural polysaccharides, these we have discussed, are the storage polysaccharide, those stored in plants and animals. The plant storage stored as starch, and the animal stores carbohydrate is glycogen. The other form of polysaccharides are structural polysaccharide, which consist the particular part of the body or particular part of the plant. Although energy storage is one important role of the polysaccharide, they are also crucial for another purpose, providing structure cellulose. For example, is a major component of plant cell walls, which are rigid structure that enclose the cells and help make lettuce and other veggies crunchy. Wood and paper are mostly made up of cellulose, and cellulose itself is made up of unbranched chain of glucose monomer linked by 1,4 glycosidic bonds. In this figure, we can see the linear structure of cellulose fibers and molecular structure of cellulose. Cellulose is made up of glucose monomer in the beta form, and this results in a chain where every other monomer is flipped upside down relative to the neighbors. Unlike amylose, cellulose is made up of glucose monomer in their beta form, and this gives its very different properties as shown in the figure above. Every other glucose monomer in the chain is flipped over the relation to its neighbor, and this result in a long, straight and non-helical chain of cellulose. This chain clusters together to form parallel bundles that are held together by hydrogen bond between hydroxyl group 4 and 5. This gives cellulose its rigidity and high tension strength, which are important to plant cells. The glycosidic linkage in cellulose can't be broken by human digestive enzymes, so humans are not able to digest cellulose. That's not to say that cellulose is not found in our diet, it just passes through as undigested insoluble fiber. However, we can say this is used as roughage in our diet. However, some herbivores such as cows, koalas, buffaloes and horses have specialized microbes that helps them process cellulose. These microbes lives in the digestive tract and break cellulose down into glucose monomer that can be used by the animal. Wood chewing termites also break down cellulose with the help of microorganisms that lives in their guts. The bee, it has an exoskeletal made up of chitin. Chitin is also a polysaccharide, and which is modified glucose units that have a nitrogenous functional group attached to them. Cellulose is specific to plants, but polysaccharides also play an important structural role in non-plant species. For instance, arthropod, insect and crustaceans, which we have seen earlier, have a hard external skeleton called as exoskeleton, which protect their softer internal body parts. This exoskeleton is made up of macromolecules, chitin, which resembles cellulose, but it made out of modified glucose units that bear a nitrogen containing functional group. Chitin is also a major component of the cell wall of fungi. which are neither animals nor plant but form a kingdom of their own. Thank you.